Assalamualaikum warahmatullahi wabarakatuh and a very good day to the examiners. My name is Syahira Amir and my metric number is A17CI0271. I am from 3SCSP. Today, I would like to present my final year project 1 with the topic of feature selection for autism classification by machine learning and statistical methods. My supervisor for this project is Dr. Zuraini Shah. First of all, I would like to briefly introduce the disease that involves in this project, which is autism. Autism is also known as Autism Spectrum Disorder, ASD. ASD is a neurodevelopmental disorder that can be characterized by the disability in social interaction and also repetitive and restrict behavior. According to Center of Disease and Prevention, CDC, ASD can happen in all ethnic, racial, and social economic groups, and there is 1 out of 54 children are diagnosed with ASD in the United States. Researchers believe that there are 2 factors of ASD, which are environmental and genetics. However, according to U 2015, genetic has at least 15% risk of developing ASD. Since genetic play a big role in developing ASD, it is important to be able to classify genes correctly in order to identify which genes is the most significant in the development of ASD. One of the data types that can be used to identify the disease is gene expression data. Gene expression data is produced by the translation process of encoded gene information into RNA. Gene expression can be used as the biomarkers to identify biological signature of autism, and it contains expression values measured from gene expression level using variety of techniques such as DNA microarray, expression vector, and fluorescent hybridization. The values is normally organized in matrix form. As good as it sounds, gene expression data has some problem that may cause biomarkers identification become harder, and that are the huge size of data, where some of data are rich and useful, while some are not. Another one is the high dimensionality of the data. High dimensionality is when the number of features or genes in the data is way more than the number of samples observed. This can lead to redundant, low quality, and noisy data, which will affect the performance when predicting important genes. Therefore, it is important to know how to deal with this problem like high dimensional in the autism gene expression data so that we can get a better performance when classifying autism. In order to do that, we need to select the most significant genes from the data. 3 research objectives has been set up for this research. The first one is to identify the computational method that can be used for gene selection in autism gene expression data. The second one is to implement the identified computational method in selected informative genes from the dataset. And finally is to evaluate the performance of classifying autism using the selected informative genes. The scopes of this research are autism gene expression data retrieved from Gene Expression Omnibus website, GEO website, with accession number GSE25507. The data contain 54,612 features with 146 samples, where 82 are autism and 64 are control. This research focuses on binary class. It implements machine learning and statistical methods for feature selection. And finally, the performance measurement measured are accuracy, precision, and recall. Next, I will summarize the findings from my literature review. The literature review starts by looking at how to deal with high dimensional in autism gene expression data. Apparently, there are 2 common ways to counter these issues, which are feature extraction and feature selection. Feature extraction transforms existing features into a lower dimensional space, while feature selection selects a subset from the existing features without a transformation. As mentioned just now, feature extraction produces a lower dimensional space by transforming the raw data. Combining, reformatting, and transforming the primary features are some of the transformations that can be done on the data. There are two categories in feature extraction. The first one is supervised, where it studies discriminative prediction for data analysis. And the second one is unsupervised, where it looks for low-dimensional space that shows the original space structure information. Linear discriminant analysis (LDA) and principal component analysis (PCA) are examples for supervised and unsupervised feature extraction respectively. Some common applications of feature extraction are image processing and natural language processing. Here are some related works on the type of data used for feature extraction. As you can see in the table, most of the data used are in image form, such as MRI and facial expression. Meanwhile, MRS data is a magnetic resonance spectroscopy, which is a time series type of data. Then we have text data, which is a text, and also microarray gene expression, which is a gene expression data. This shows that feature extraction are very common when it comes to extracting image or time series type of data, as well as text. On the other hand, feature selection identifies and selects significant features from original data into a subset without transforming the data. Feature selection will decrease the number of features to sample ratio, and it also reduces computational load. Thus, it will improve the prediction performance. There are three categories in feature selection. The first one is filter, the second one is wrapper, and the third one is embedded. Filter feature selection uses feature ranking techniques as the basis of gene selection, where top rank are chosen as the informative features. One of the examples of filter is t-test. The common assumption in t-test is the values of genes in two compared groups are equally distributed. T-test null hypothesis assumes equal variance and equal means. Alternative hypothesis reject the assumption. T-test result in 0 or 1 output. 0 accept null hypothesis at 0.05 significance level, while 1 reject the null hypothesis at 0.05 significance level. T-test will also return p-value, where small p-value signifies significant difference among the compared features. Based on Jovic et al. 2015, the advantage of t-test is it gives base accuracy performance in the classification of gene expression, and it also fast and simple method. Next is wrapper. Wrapper evaluates feature subsets based on the performance of classifier. It takes model hypotheses into account by testing and training in the search space. One of the wrapper examples is genetic algorithm. Genetic algorithm is a randomized wrapper that produces ranking of features. In genetic algorithm, the search space elements are either arrays or binary strings. Genetic algorithm works by generating new population strings iteratively from the old strings. The advantage of genetic algorithm is it is able to find optimal solution among the search space by using genetic operators. The common genetic operators used are mutation, selection, and also crossover on the initial random population. The last category of feature selection is embedded. Embedded performed during the execution of modeling algorithm, where it embedded in the algorithm either as extended functionality or as its normal. It is a classifier dependent, where the selection influenced by the hypothesis made from the classifier when the ideal set of features is developed. One of the examples of embedded is random forest. Random forest returns several variable importance measures. These variable importance measures is based on the classification accuracy with values of variable in a node of tree that are permuted randomly. Embedded random forest works by build new forests at each iteration after discarding features with the smallest variable importance. The genes with the smallest OOB error rate are being selected as the significant features. The advantage of random forest is it is faster to build and predict, and it also performs well in finding and classifying useful subsets of genes. Next is feature selection related works based on the type of data. As you can see in the table here, microarray gene expression seems to be one of the most commonly used when it comes to selecting the most significant features by using feature selection. There are 4 research that are using microarray gene expression with 2 different types of disease, which are cancer and autism. However, there is only one type of data that are not using microarray gene expression in their research based on the literature review that I'm doing here, which is the research use text data. So from this table, it shows that microarray gene expression work well with feature selection method. After I finish with literature review, I've decided to choose feature selection as the method to select the most significant genes or feature for my gene expression data. This is because it is commonly applied on gene expression data type. It is also very effective in selecting useful genes. Feature selection gives high accuracy in classification, and it also yields stable result, which will help me to achieve the research objective in the future. Next is the research methodology. There are 3 phases in research methodology, which are Phase 1, Phase 2, and Phase 3. Phase 1 is literature review phase. The aim of this phase is to study and collect information from related resources, and also to identify the goals, objectives, and research scopes. The next phase is Phase 2, which is implementation and development. The aim of this phase is to identify dataset and algorithm to use, to do pre-processing on the dataset such as data reduction, and also to implement the statistical and machine learning methods on the dataset. Finally, Phase 3, which is the result, evaluation, and analysis phase. This phase is aimed to evaluate performance of gene selection methods and then discuss the result and concludes the research conducted. Next is the performance measurement. The performance measurement used to justify the result of gene selection by classifying the selected genes. The classification performance is evaluated based on the accuracy, precision, and recall, which is formulated from the true condition table shown in Table 3.1. Equation 3.1, 3.2, and 3.3 is equation for accuracy, precision, and recall respectively. This equation is based on the true condition table. Now we will go to research design and implementation. Here is the flow of the proposed solution. The proposed solution starts with data collection from georepository, then proceed with data normalization using RMA and AMS 5.0. After that, data reduction using Geo2R tools that produce top 250 features. These top 250 features will be used for pre-selection by filter, which is a statistical method called t-test, and will produce filtered features. These filtered features will be input for the machine learning algorithm. There are 3 different machine learning algorithms that will be used in this research, which are SVM-RFE, random forest, and also genetic algorithm. By combining the filtered features with this machine learning algorithm, it will produce 3 optimal features separately, which are optimal features 1, optimal features 2, and optimal features 3. After getting the output, this output will be evaluated by classifier and through the accuracy, precision, and also the recall result. Data collection is where the data set is being retrieved from georepository using accession number GSA25507. The first time the data being retrieved is in text format, and after that I open and transfer it into Microsoft Excel and then save it into CSV format. The reasons I choose to save it in CSV format is because it is easy to incorporate in many analysis tools and also programming languages such as Weka, Python, and MATLAB. On the right side, shows the figure 4.3, which is Autism gene expression dataset that is being saved in CSV format. On the rightmost column shows the ID of the genes, which is the genes ID, and on the top row is the sample ID, and in the middle is the value of gene expression based on the ID genes and the samples. As for data normalization, the data has been normalized by the data contributor, which is Alter et al. 2011. According to Alter et al. 2011, data normalization is done to balance the individual hybridization intensities by adjusting it appropriately. And it is also to create a meaningful biological comparison. The method that is used for data normalization was Robust Multi-Array Analysis, RMA, and also Microarray Analysis Suite, MAS 5.0. MAS 5.0 is used to analyze the gene expression distribution, while RMA is used for quantile normalization, and it has a potential in removing group-level differences in the distribution by looking at specific differences between the groups. The significance threshold in normalizing the data is p-value equal to 0.05 and the fold change of 1.1 fold. All data has been checked and they have similar distribution pattern. After that is data reduction. Data reduction is the process that is done to reduce the number of features into considerate number. Since we are dealing with a huge amount of features, which is around 54,000 features, we need to reduce it in the considerate number to make our analysis more practical and efficient. So the tools that I use to reduce the data is called Geo2R. Geo2R compares two or more groups of sample, which in this case is autism and control groups, and then it identifies the differentially expressed genes, which is DEGs, and after identifying it, it will select top 250 genes from the samples. On figure 4.4 shows the defining group samples in Geo2R. The blue color is the control group and the pink color is the autism group. The data reduction by Geo2R use p-value less than 0.01. It also uses Benjamini and Hochberg false discovery rate. The reasons why it used this Benjamini and Hochberg false discovery rate is because it is most frequently used and it yields a balance between the limitation of false positives and the discovery of statistically significant genes. Geo2R also apply log transformation on the data. And after consider everything, it will produce the top 250 based on the p-value result. The smallest p-value genes, meaning that it is most significant. As you can see in the figure on the right side, features 235,934_80 has the most significant features with p-value of 0.00000094, which is the smallest p-value. The output result from data reduction, which is the top 250 genes, will be used for feature pre-selection by filter, which is a statistical method called t-test. And this t-test will produce output called filtered features. And the filtered features will be used on 3 different machine learning algorithms, as mentioned before. And then these 3 algorithms will each produce optimal features that will then be used for evaluation by classifier that will look at the accuracy, precision, and recall. This whole part will be done in the future, which is in final year project 2. In conclusion, the first objective, which is to identify the computational method for gene selection, has been successfully achieved. The computational method that has been identified is feature selection, which are statistical method, filter t-test, and also machine learning method, which are wrapper, SVM, RFE, and genetic algorithm, as well as embedded, which is random forest. For the future work, I will work on achieving the other two objectives, which are to implement the identified methods and also to evaluate the performance based on the accuracy, precision, and also the recall. That's all from me. Thank you for listening and have a nice day.